I'm delighted to introduce our first presenter, who is Camphire, whose presentation is titled Bacterial Infection or Protection, Exploring a Microbiome Checkpoint.
If you owned a bank, in particular a bank that was known to be the target of bank robbers, you would want the best security possible. You would want a team that was positioned at the site of the break-in, was able to detect an intruder when they arrived, and had the means to stop them before they could do any harm. You probably don't actually own a bank. I don't either. But all of us are in this situation right now. And I think some of us have better security than others. I'm a microbiologist, and I study the bacteria Streptococcus pneumoniae, or pneumo for short. Pneumo is a major problem. And unfortunately, we are the big heist. We are the bank that this bacteria is constantly trying to break into because it cannot survive unless it colonizes a human. Extra bad, this is the third deadliest bacteria to humans globally. This heist has huge stakes. But as deadly as this bacteria can be, it actually doesn't always cause disease. Before it ever makes us sick, pneumo first colonizes our upper airway. And here, it does not cause any illness, making this initial colonization stage a sort of checkpoint before pneumo can do any harm. But why doesn't pneumo always make it through this checkpoint and cause disease? This is actually a big mystery in my field, and a question I'm addressing in my thesis. My hypothesis is we have a member of our microbiome that meets all of these criteria for security, and by filling this role, provides protection and prevents invasive infection. So who is this candidate security agent? I would love to introduce you. Their name is Streptococcus mitis, or mitis for short. You may not know mitis, but they know you. Almost everyone in this room right now and watching on the live stream has mitis in their upper airway right now. And this is the same location that pneumo tries to colonize, already positioning it at the scene of the break-in. So now all pneumo has to do is detect our intruder and stop them before they can pull off this heist. But how? In my thesis, I analyzed several thousand genomes to understand a specific tool that mitis has to get this job done, as well as what makes that tool succeed or fail. The tool that mitis has is a receptor, or a sensor of sorts, for this tiny chemical signal that pneumo exports while it's colonizing our upper airway. That signal is that orange squiggle up there. My work suggests that mitis is able to detect the signal. When this happens, mitis gets activated, and it can mount an attack in response, stopping our intruder and stopping the robbery in its tracks. But unfortunately, I said the tool can fail sometimes as well. My work also showed that sometimes the signal from pneumo and the receptor from mitis just don't match up correctly. And that gives us the situation in the bottom row here. Pneumo's making a signal, but is undetected by mitis. Pneumo can now just run past our guard and cause disease. I would love to be able to tell you that the situation in the top row happens far more often than the bottom row, but we just don't know enough yet. As I continue my work, I hope to demonstrate exactly whether and how mitis stops pneumo at this checkpoint before disease, and ultimately describe how we can leverage our microbiome and use its tools, not just our own, to help bring us from infection to protection. Thanks.